we just started new series which are from Sindh Accdemy Umerkot MCQs. Till last video we have completed MCQs till 780 somewhat. This is simply the grand test. We have taken screenshot and I'm explaining each one for your understanding. So let's go. All the following are directly involved in mitochondrial energy production except. So mitochondrial structure, I hope it's clear. I've repeat multiple times. It simply says inner membrane, outer membrane, some crystal like structure. So DNA, only DNA is not part of it because DNA is simply a genetic material, deoxyribonucleic acid, which have some nitrogenous bases, phosphate, ribose, sugar, and these all. So this means cytochrome, yeah, cytochrome B complex is present in the mitochondria. NAD, ATPase complex is present for ETC, Krebs cycle. So C is the right option, which says DNA, because they are except not involved in mitochondrion, that's DNA. The thermophilic enzyme preferred in high temperature biological washing powder because they resist denaturation and maintain activity. So yeah, thermophilic enzyme. Thermophilic means temperature, like that. Thermo, wherever you word see it, it means it's related to temperature. So preferred in high temperature because biological washing powders, because they resist, this means they help them resist denaturation and maintain their activity. The face of the Golgi complex, that is convex and oriented towards the nucleus. So we have Golgi, which helps in the modification, some modification and transportation, making their structure. So their structure is different. For suppose if I could make their structure, this is this kind of a structure. So here, for suppose we have nucleus, so near the towards the nucleus, this means the in the this convex type structure is called as cis phase, and their structure like these folds are called as, in mitochondria we have cristae, but here it's called as cisternae. Okay, cisternae. And the entire this region is called as lumen. Lumen simply means cavity. So here this structure is called as transphase, which is concave, but this is cis phase, which means it's convex. So this means B is the right option. Pancreatic amylase works best at 8.5. The 70s ribosome in mitochondria propose an evolutionary link to 70s. We have 70s or 80s. We say S is simply Swedberg unit. This have subunits which are called as like 50 and 30. Now if we add them it become 80, but we cannot add them, it's simply their units. So prokaryote, which has 78, 88 is for eukaryotes. This have some subunits called as 60 and 40. So it's not supposed to add, but it's just learn it. 88 is for eukaryotes, 78 is for prokaryotes. Second question, the correct statement about telocentric chromosome. So we have different types of chromosome like this. If it's at the middle like proper shape, it's called as metacentric. Then submetacentric is simply chromosome become a little higher than the normal position. Then comes third one, which is about metacentric, submetacentric, acrocentric, a little higher. But in the telomere stage, but centromere comes at the top, but I mean comes at the end. Okay, so centromere is at the end. This is the right option. Then let's say mitochondria can divide individually because mitochondria they have their own genetic system. Same goes for chloroplasts. Why? Because they do follow endosymbiotic theory, which was given by Mylan's kind of scientist. Find the right set of activators. So we have activators like manganese, magnesium, chlorine. These are the right option. A structure present in eukaryotes but absent in prokaryotes. So eukaryotic cell have nucleolus which synthesize ribosomes. At very high concentration, reaction rate might slow, or it called as retard, because when high structure is there, reaction gets slowed down because we need enzyme even to speed up. The structure liable for molecule transport membrane is called as nuclear pores, because these are only present for the molecule transport. Prokaryotic cell have genetic material which is prokaryotic has kind of DNA, circular DNA without histone protein, but in eukaryote they have some linear DNA with histone protein. Together they form a structure called as nucleosome. In chlorophyll, cofactor is called as manganese containing porphyrin. Yeah, this is the right option. Which chromosome has equal arms? If we see their shape, the first one called as metacentric, which have both equal arms. Submetacentric have a little upward telocentrin, where centromere comes at the end. Acrocentrin is the third place, where you'll see a little like lower arms are more. So this mean here A is the right, C is the right option. What happens to the reaction rate at as substrate concentration increase? So rate increase as well, because reaction substrate concentration increasing with constant enzyme. This means substrate is also provided and enzymes are also provided. So this means rate increase. None of them is going to be limited at that region. Sister chromatin are linked together. If we see, this is a chromosome. This is one chain. This is one chain. Both together called as sister chromatin. And for suppose if this and these both are connected, then these are called as non-sister chromatin. So this means right option here, they are connected by centromere. The charge and shape of the active site of enzymes are defined by amino acid and polypeptide chain. Okay, the enzymes found in nucleoplasm. So enzymes in nucleoplasm are called as RNA polymerase. ATP synthesis in mitochondria. Amylase is in mouth to break down starch. Maltose catalyst is also different. Which type of inhibition involved binding to the enzyme substrate? ES complex. So inhibition which ES complex, that's simply uncompetitive, which is also called as anti-competitive, where they inhibition involve binding enzyme substrate, neither they do bind with the allosteric site nor to the active site. Okay, then the molecule which are metabolized from mitochondria ATP is our carbohydrate, lipid, protein. Molecule which are metabolized by mitochondria which provide them ATP, it's carbohydrate, lipid, protein to provide them energy and all. After catalyzing a reaction, enzymes are consumed. Catalyzing reaction, enzymes are decomposed, enzymes remain unchanged. So yeah, this is the right option, because until unless we provide them substrate and enzyme both, it becomes same. And the feature that distinguish induced fit model and log key model is simply the flexibility of active site during catalysis. Yeah, lock and key model was given by Emil Fisher, induced fit model was given by Koshland. These both are opposite kind of models which were given. Very important. I hope you know this, and I've already told in like multiple times in the previous videos of this series. All the following structure are part of Golgi complex except ribosome itself is a new organelle which helps in protein synthesis. It itself is synthesized by nucleolus in prokaryotes. So cristae is simply a folding which is present in Golgi vesicle, tubules, all of the part of Golgi complex. Penicillin is an example of competitive inhibition. Learn this. This means it get attached uh, to the substrate, and active this attached to the active side. Non-competitive means attaching to the allosteric side. Uncompetitive means attaching to the ES complex only, which nothing like this happens. It is a competitive. The chromatin network break down into chromosome during cell division. The fluid filled substance inside the nucleus is called as ANC. This means karyolymph. And nucleoplasm, yes, fluid filled inside nucleus. So nucleus this, but if we talk about the cell, then that structure is called as cytoplasm. So I hope it's very clear. We have covered everything in so detail. These all of the MCQs taken from the Academy, some screenshots to just make you understand. I hope you like it.